computer. And then I'm going to share my screen. Okay, so you can see my screen. Let's get rid of this. Okay, so this is the web page in Canvas. This is the current version of the syllabus out there. I'm going to change some things today, tomorrow. We went over this material last week on I'm just realizing that if she tries to enter, I can't see that she's trying to enter. Let me stop for a second. That's a defect in the system. I can't see that she's trying. Well, I'll have to go back and forth and check on her. Okay. So what we did by way of making sure that this was going to work last week, along with the video recording and how I was going to store it, etc. So I did a review of a pH lecture I give to medical biochemistry. I went through some stuff in this pH handout. And then I assigned you problems out of Siegel. And the idea was that in those 3 exercises, especially the work you did in the assignment, this would sort of bring everybody up to speed and we would be at the same place, okay? So what we're going to talk about today, I'm going to do a little introduction to set up a context, and I'll use a PowerPoint that I haven't posted yet. It's nothing but mostly stolen slides from these 2 papers, or stolen information from these 2 papers. But we're going to cover these 2 papers, this 1966 biochemistry paper and this 1976 biochemical journal paper. Okay. I'll allude to other stuff that will come up later as we set the stage for other things. And then next week, we're going to get into something involving statistics. Okay, and I'll tell you more about that then. So is Cindy showing up yet? No, she's not showing up. Wow. Okay. So one of the things, let me just say this at this point. I don't think I said this last week, and I don't know that I say this to every class in exactly this way. It'll be obvious to you as we go along that this course is not done as a single book. I think I used to teach out of a single book in the 90s. And we will use chapters out of Van Hold's Biophysical Chemistry. But I got away from that because I like covering a lot of papers. They allow me to do some things that I couldn't do with a book that I want to do. Okay. So I'm giving you a blend of manuscripts, papers, book chapters, etc. And the reason is that I'm trying to teach at a slightly higher level than what a book might do for you. And I'm trying to make things a little more practical than a book alone might allow it to be. And at the same time, I'm also trying to get you used to an idea. And the idea is that your education from this day forward, every day forward, depends on what you put into it, depends on what you read and what you think about and what you explore. And if you're limited to the, the kind of straightforward stuff that people do in a book, which doesn't I'm not complaining about textbooks, but I'm saying that they're sort of limited and straightforward. Jump this hoop, learn this material, you can move on. I want you to get used to the fact that there's a lot of stuff out there to know and understand. And the only way to do that is to constantly be reading papers. Primarily, you're going to be doing that within the context of your own research, but you really have to be constantly exploring beyond that. And I do that constantly in my own work, in my multiple projects and collaborations, and you need to be doing it too. I'm doing it as I teach. I'm constantly reading things and thinking about presenting things, okay? So Cindy is still not showing up, okay. So for example, in the syllabus, after we do statistics here, and these are 2 chapters out of this textbook by Bevington. This will be on error propagation, and this will be on linear least squares. I would generally go into a series of chapters on thermodynamics that come out of past versions of Van Holt's book, but I'm pretty sure I'm going to insert something right here on the whatever the next date is after the 25th, so it's probably February 1st, and we're going to cover a chapter out of a book that I almost never teach out of, but I would like to teach out of, and John Hassler actually wants me to cover this chapter too. So it's a chapter in Creighton's book. I think he has it as a reference on his webpage on the Canvas site for him. I think it's chapter 4 out of some version of Creighton. He may have a different version than the one I'm going to cover, but it covers the same material. And at some point in here, I'll also cover a series of lectures that are indicated here in the syllabus. You can't see this because I don't think I've shared it with you, but on my version, I have something called hydropathy. And in that hydropathy chapter, I have a series of papers that we're going to cover. So there's the Creighton chapter I'm going to cover. But then there's a series of papers that have something to do with hydropathy. So it'll be a method by Kite and Doolittle that you may be familiar with because John probably talked about it in Med Biochem. It has to do with predicting transmembrane helices. And then a chapter by Steve White, a paper by Steve White on interactions with membranes, protein interactions with membranes. And then I think this is a paper on prediction of membrane protein embedding in membranes. And so it's introducing some things that kind of brings together protein structure, but also some biophysical concepts. And so I'm going to do stuff like that because I like to learn things myself. And so the best way for me to do it is to give a new lecture. I'm doing it in part because I'm working on a project with some collaborators on hydropathy and predictions, and I'll tell you a little bit about that when I give this lecture. Okay. So let me bury that. Let me bring this up. Okay, so this is the PowerPoint that I haven't yet posted because I was playing with this on Saturday and yesterday. And as I say, I'm just putting stuff into it out of the papers, but I need an introduction here. Sorry, I'm going to go back one more time to check on Cindy, and she's still not showing up. Okay. So these are the 2 papers that we're going to talk about. Let's get this off to the side a little bit. This paper by Good. I will refer to this as Good's buffers because the paper is about hydrogen ion buffers for biology. And then this paper by Storer, Cornish, Bowden, which has something to do with magnesium ATP, and I'll give you that context in a second. So this is one of the the slides that we talked about last week. It's out of that pH lecture, and it shows a titration curve for acetic acid. As you add base, you convert from the acid form to the basic form, and you get this curve. The figure, I don't think I pointed to, I know I didn't point this out last week. The figure highlights what is called the buffering region, this red highlighted region. And the red highlighted region corresponds to a region where the buffer is between 10 and 90% deprotonated. Okay, and so down here, approximately, that's at 10%. So in the Henderson-Hasselbach equation, let's see if I can get this to work. In the Henderson-Hasselbach equation, it would look something like this, or it's the log of the base, and I'll just call this 0.1. And the acid, let's call it 0.9. Keep. Oh, goodness, I didn't want to do that. Sorry. And it might, maybe what I actually want this to do is 1/10. Let's just do 1/10. Don't worry about all of that. Okay. And so the log of 1/10 is -1. So the pH will be -1 below the pK. So it'll be something here approximately at 3.8. Okay. And then at the other extreme, it will be 10 parts base, 1 part acid. And so the log of 1, and so the pK, it will be 5.8 instead of 4.8, the midpoint. And so it's the buffering region because it's between 10 and 90% protonated. All right. And that property has to do with the fact that acetic acid has a certain affinity for a proton. And so the minus the log of the K for the loss of the proton will give you this 4.8 value, and it will give you this feature. In other words, the chemical property of the group. Okay. Now we show another figure in that involving alanine, and it's showing the fact that there are 2 protonatable groups, one with a pK of 2.3 that corresponds to the carboxyl here. Yep, sorry. And one that corresponds to the amine. Okay, and so there's a successive loss of a proton when you titrate. But the point is that this group, the carboxyl, will act as a buffer around pH 2.3. And this group, the alpha amine, will act as a buffer around pH 9.1. So there are 2 buffering regions, and each one of these has what we would call a 10% to 90% region. It's usually the pK plus and minus 1. And this, again, also here has a pH region approximately 10%, 90% around the pK. So you could write the same sort of pH, pK region equation I just wrote down, but it would be with a pK of 2.3 and a pK of 9.1. So these would be buffering regions. And you could use alanine to buffer a solution at pH 2.3 and a pH of 9.1. All right. So that's the idea of the buffering region. Here you're adding base to force the reaction to a particular pH. But generally speaking, the idea is that the buffer in a solution keeps the pH at some stable pH. And it becomes important when you have a reacting system that might be generating protons or using up protons. Most of us don't use dynamic systems in our in vitro experiments. In cell culture, for example, there's all kinds of reactions going on and there's acid being produced. And so there are buffers that are in the media, and you use some kind of a marker to indicate that the pH is being buffered in your solution. So you're replacing the media, for example, and partially what you're doing when you're replacing media is replacing the buffering capacity. Okay, let's go back one more time. Sorry, still no Cindy. Okay, that's a bug in this system that I can't see people. What I intended was that you guys would just be able to enter with the password. And the password is obviously to keep problems out, but I thought it would allow you in. And so I'm going to have to fix something. Okay, so this is a titration of histidine, which has 3 protonatable groups. Okay, and so you can see there's a protonatable group corresponding to the carboxyl. And so the buffering region here would correspond to this region between approximately here and here. And then this is a pH titration region corresponding to the imidazole. And then this is a pH buffering region corresponding to the alpha amino. So there are 3 buffering regions for histidine. And the way I generated this curve with a pK here, I'm obviously choosing something around here as the pK of the imidazole, okay, which may be a little bit higher than what a textbook might say. So 3 buffering regions, okay? And so histidine could be used as a buffer around 2.5, around 6.5, 7, and around 9, okay? And then there are lots of other questions you could ask about this. In this particular case, off of this example from past exams, what's the isoelectric point? And down here at the beginning, the imidazole is protonated. So down here, we start out with a +2, and then we lose the charge. We generate a charge, excuse me, when we deprotonate the carboxyl. And now the charge on the protein is +1. And then it's not until you get over here that you tend to get to something that's close to 0 as you lose the imidazole pK, okay, and you have a plus and a minus, and so the isoelectric point ends up being somewhere in the middle between these 2 reactions, because this takes you towards 0 over here as you lose the 3rd titratable group, okay? So it's somewhere in between. And so this again highlights the different buffering regions, the different kinds of questions I could ask. In medical biochemistry, you spend a lot of time talking about the fact that you needed to know the structure as it changed through these different stages. I won't go over that right now. Okay. Here's another kind of question that I've asked in the past. I was looking for it in past exams, and I mustn't have asked this question in a long time. Which of the following amino acids can serve as a buffer at pH 3? Okay. So what this would be asking is, what does the titration curve look like? For example, for alanine, that's what the A corresponds to. And so you would have something that looks like this, and then something that looks like this. And these would be the buffering regions around here and around here. And so alanine could serve as this. But what would the titration curve look like for a glycine? And the answer is it would actually look very similar, if not identical. And so glycine could also serve as a buffer at pH 2. Okay. And then glutamic acid and arginine and histidine all could serve as buffers here, even though the histidine would have another group, and then it would have a 3rd group up here. So I drew this at the wrong scale, but we would have something that goes up to a 3rd titratable group. Okay, the glutamic acid would have one here, and then it would have a 3rd group, but it could buffer at pH 2 or 3. Okay, and the arginine would have a curve that sort of looks like this, but then it goes up for another curve out here because of the side chain of the arginine. So all of these amino acids in principle could be buffers at pH 2. And so the trick to this question was simply recognizing that they all have a carboxyl. They could all be buffers. Some of them could buffer in a wider acidic pH region. Some of them could buffer in a wider basic region. Only histidine could buffer around pH 6 or 7. Etc. Okay. Again, the principle is the functional group, and the affinity of the proton for that functional group is what dictates the pH region. And I didn't write this down, but this is pH down here, and this is equivalence 1, 2, and 3 equivalents of protons titrated. Okay, so that's the idea. Okay, so this is a summary of stuff that's coming. This comes directly from the goods buffer paper, and I'll switch to it in a second. The choice of buffer is a critical aspect of doing biochemistry. And so what I mean by that is you want to use a certain pH for some reason, okay? Generally speaking, we don't do biology at pH 2, and we don't tend to do biology at pH 9 or 10. So those groups usually don't help us in vitro. We tend to do biology between pH 6 and 8. And so we're looking for things that are titratable in the range of pH 6 and 8. Okay. The problem is that in the 60s, when this work was being done, there weren't a lot of buffers at pH 6 to 8. The primary one was phosphate. And phosphate, the second loss of a proton for phosphate is a pK of 6.7. Okay. But generally beyond that, there weren't a lot of things. Okay. And so generally speaking, when we choose a buffer, we're sort of mimicking what other people did. We're told, well, this is what they did when they studied this, so this is what you're going to do. And then we play around with various pHs. I can't use this. We play around with various pHs. We vary the salt concentration. We'll talk a lot more about that. We talk about the concentration. We also talk about the identity of the salt. Okay, are we using sodium? Are we using chloride? Are we using potassium? What are we using? Okay. We play a lot with things like temperature, reducing agents, detergents, et cetera, but that's part of the whole issue. Okay, and so having a condition that sort of makes sense is really what we're sort of focused on. Okay. And so what this paper is about is the fact that many of the buffers, many of the buffers have unusual properties that for various reasons don't work in every system. So for example, phosphate does not have buffering capacity. It's beyond the 90% capacity, up around pH 7.5. It also has a tendency to precipitate certain kinds of anions, cations. It is often a metabolite. Anytime you talk about ATP phosphorylation, you're cleaving an ATP. The phosphate is a product of a reaction, but that phosphate could also be an inhibitor of many of these reactions. And so you have to be careful. Tris has a high pK, so it tends to be best in basic conditions. But it also can be reactive and inhibitory in certain kinds of reactions. And then other kinds of buffers, things that you're probably not very familiar with, things like borate or glycyl glycine. Generally speaking, they have issues that make them inappropriate. Okay. Veronal is actually referring to a barbiturate buffer that almost no one really uses anymore. So a lot of these are just very unusual buffers, and people don't usually tend to use them. Okay, and so what these guys set out to do was design a new series of buffers. And so let me actually switch out of this and say discard that for the moment. And let me come over here. So this is the paper, and a lot of the notes that I was going through come directly out of the paper. So if you've already looked at the paper, you already sort of know what I'm saying. Okay, so the abstract is describing the classes that we're talking about, and this introduction here describes all of the things that I just said. I think it's actually a copy paste of what's in this initial section here, describing the problems with many buffers and the fact that it kind of limits what we can do. All right, so the question is, what would be the kinds of features that you would like to have for a new buffer? Let's go check on Cindy again, and she's still not showing up. Okay. And so they have a series of rules. I know you have access to this on your own computer, so you can see this. Let's just make it bigger. Okay. So the first rule is this feature that we would like to have buffers that have pKs between 6 and 8, because a lot of biology happens between 6 and 8. Now, there's some biology, for example, if you were studying the stomach, you know, and what happens in digestion, then obviously things can be much more acidic. But generally speaking, cells, biological fluids tend to have mostly neutral pH regions. And so you would like buffers that have pKs around 7, but you would also like them to not have sort of inhibitory properties that some of these other buffers that are described in the paper might, okay? The second feature is that you would like the buffer to have good solubility in water. It says minimal solubility in other solvents. This gets into discussions about membranes, unusual non-aqueous solutions that are usually very unreasonable. Generally speaking, buffers are charged, or one form of the buffer is charged. We'll talk about that in a second. But generally, solubility is one of the things that you would like to get into, and I'll say something about that when we get to the list. Okay. Okay. Concentrated stocks is the primary issue. In other words, when you make up a buffer, and it may be that none of you make up buffers. It may be that your lab buys the buffers. Okay. And that's generally true. My lab tends to make up buffers, although I've started buying some stuff recently. And so you want to make up a stock. So even if you were just making up PBS, you would generally make up a stock at a higher concentration so that you could do tenfold dilutions, and you could have it around, and then you could dilute it down to working conditions without having to make it up every day or every week or whatever. Okay. And so you need the solubility to be reasonably high so that you can have concentrated stocks. Okay. Tenfold is sort of a target, but it depends on what you're working on. Okay. All right. And then the problem, therefore, is you would like that stock to be soluble. All right. As I say, I'll give you examples in a few moments. Okay. So that would be one of the criteria. A third criteria has to do with not passing through membranes. Now, this is not typically an in vitro problem. This is generally a cellular problem, depending on what you're working on. We did a problem at the end of that lecture last week about transport across a membrane. I don't think we went over it in class, but it was there. Oh, no, we did. We talked about aspirin and what fraction of the group was uncharged and could passively diffuse from the stomach into the bloodstream. And then we went on to say from the bloodstream into, for example, an organelle where it might actually inhibit, okay, whatever its target is. And so these features are things that you sort of would like to think about. This is a little bit simplistic because usually buffers change their charge. Okay, they change their charge when they get deprotonated or protonated. And so it's one form or the other that can't cross a membrane. Okay. So that's one of the features that we're going to be looking for. The buffer should produce a minimum of salt effects. Now, this is an interesting issue, and we'll come to that and when we get going into this. What that basically means is you're generally adding a buffer. Let's say, let's write down here. Let's say you make something up that's 100 millimolar phosphate, okay? pH 7, okay? So this will necessarily be a mixture of two species, okay? Because there's a deprotonation that goes on between these two species. And so these will be the species in solution, okay? And these will be the charges on these species. And then you will generally also have something else in the solution, 150 millimolar sodium chloride. Often when people make up PBS, you actually use some mixture of potassium and sodium phosphate. Okay, so you use some mixture that gives you a combination of species. Okay, and so you also have potassium often in the solution. And so that's what they're referring to here. They're referring to salt effects. What they're really referring to is the net charge on all the charged species. They're essentially referring to what is in that section in Siegel as the ionic strength, where the ionic strength is a function of the charge squared times the concentration summed over all charged species in the solution. In this case, it's summed over the sodium, the potassium, the phosphate moieties, the chloride moiety, okay? And so it's this ionic strength that we're usually talking about, all right? And we'll talk about why that is in much more detail as we go along. But basically, it's charge-charge screening. It's the salt causing effects because the ionic strength is so large, the charge is so large in the solution that it might inhibit interactions between charged-charged species, or it might change solubility of certain things. We'll come to this in that Creighton chapter that I talked about. There will be conditions where certain kinds of salts will precipitate proteins. Okay, and so you don't want that necessarily to happen unless you really want it to happen on purpose, if you're trying to fractionate things. So ammonium sulfate is often used for fractionation. And as you raise the concentration of ammonium sulfate and as you drop the temperature, proteins will precipitate. And they will precipitate selectively. Some will and some won't, and you could use it as a purification method. But you don't want it happening to your protein while you're working on it. And so you want to be careful about the salt concentration or the salt effects of the buffer that you're using. For that reason, very often, buffer concentrations are usually kept small, okay, so that there isn't a lot of buffer effects, but then you put in a lot of salt for keeping things in solution, but not having lots of charge-charge interactions in that solution, okay? We'll come back to this in a number of different ways. Okay, number 5 has to do with the concentration, temperature, ionic composition of the medium and the dissociation of the buffer. This continues this same discussion that basically these other solvents that you have do or don't influence the buffer itself. Buffers might precipitate. One of the buffers that we're going to be talking about is pipes. Okay, and when you make up pipes, you dump it into solution. So I usually would make up a 400 millimolar stock. Okay, so I would weigh out enough pipes in a 2-liter container to make it 400 millimolar. It's an acid when I add it, so it dissociates in the same way that any weak acid would dissociate, and it makes an acidic solution. As soon as that happens, pipes precipitates. It is insoluble at pH 2 or 3. You have to titrate it up to the pH you want. I usually use it around pH 6.9. And so I have to titrate it. I have to add sodium hydroxide to titrate the pH up to 6.9. Okay. And so what this is saying is that there might be other things, including the temperature, because the solubility is temperature dependent, or other kinds of components that might be in solution that would cause precipitation. You don't want that to happen. And so that's what this is really referring to. Okay, so complexes formed with cations. And so let's scroll this down, sorry. And the binding constants of those cations should be constant, should be known. Some of the buffers should not make such complexes. Okay, so sometimes that's not controllable, but that's part of the issue that you're sort of dealing with. You would like to sort of know what the buffer interacts with. All right, the second paper we talk about, we'll go into this in greater detail. Okay, but you would also need to sort of know what all the binding constants are. And there are sources for this kind of stuff. Any buffer that you might use or somebody might suggest you use, you can find information about the buffer in the fact sheets that usually come with the buffers when you buy them. Okay, the buffers should be as stable as possible. They should resist degradation, some kind of inhibition in particular enzyme reactions. Occasionally, buffers are reactive. Phosphate is the good example. There's a discussion at the beginning of this paper about tris. Okay, and so these issues are things that you should be aware of in the chemistry that you're involved with. They should not absorb light in the visible or ultraviolet. Now, the reason for that has to do with protein concentrations primarily, but it could be for DNA concentrations as well. You know, I talked to you about this a little bit, and we'll say more about this, that most proteins tend to have a UV spectra. So this is wavelength. This is absorbance here. And most protein spectra sort of look like this, and then they come like this. Okay, sorry. And this is 280 usually. This minima usually is around, sorry, something's getting in my way here. This minima is usually around 255. There's usually another, there's sort of two peaks in here, one around 276 and one around 280. The 280 one usually corresponds to tryptophan. The 276 corresponds to tyrosine. This peak is therefore due to the sum of tryptophan and tyrosine. We'll talk about this in more detail in a further lecture. If you have DNA in here, then your peak tends to be around 260. Okay, and so you get your concentration from a 260-ish peak. It's not exactly 260. It depends on the composition. Adenine, thymidine, guanosine, cytosine, they all have slightly different maxima, but generally that's where it comes from. Now, if your buffer also absorbs in this region, okay, then it in principle can interfere with this signal. Okay, now generally speaking, you blank to do a spectra to get a concentration, but buffers like pipes and buffers like MES tend to have fairly high absorbance. It primarily is down here in the lower wavelength range, in the kind of 220 region, but generally speaking, they will add noise to your signal. Okay, there are certain other spectroscopic techniques that we talk about. We'll talk about circular dichroism later in this course, where we go all the way down to 190 or lower, and these buffers become terrible down there. Okay, this is an advantage of phosphate. Phosphate has no absorbance all the way down to 190, and so phosphate is a great buffer spectroscopically. But buffers like pipes or MES, or for that matter, tris or HEPIs, they all absorb in this kind of region, and so they add noise to the spectra. Now, a lot of you probably are using simple devices to make concentration measurements. I know there's this nanodrop thing that I've actually never used in my life. And you push a button and it registers an absorbance and it gives you a 260 or it gives you a 280. But the question that I would have is, how is it blanked? Okay, are you using the buffer to blank it so that you get a baseline? Because the buffers can give fairly high numbers, fairly high values. I'll talk about this a little bit later because I know a lot of you are not familiar with techniques like dialysis and equilibration of samples, and how you would go about that and why you would do it. And one of the reasons for doing it is the fact that the buffer gets in the way. And so you want to make sure that it doesn't interfere with your concentration determination. Noise is part of the issue. It just makes the signal noisy if you're trying to observe something at 260 or 280. Okay. That's number 8. Number 9 is they should be easily prepared from inexpensive materials and should be easily purified. Okay, so this is a 1966 concept. 1966. And the reason I say that is these people were actually making these in the lab. And I'm going to go back and see if Cindy has tried, and she hasn't tried. Okay. You would never do this today. Okay. This is a foreign concept to you. In the 60s, people made their own pH meters. They made their own electrodes. You couldn't just go to a store like Fisher and buy stuff. Okay. It sort of didn't exist. This is a foreign concept today. You just go buy pipes or mess or phosphate or tris or HEPIs, etc. Back in the day, it wasn't that easy. Okay, and that's part of what this paper was sort of all about. So number 9 doesn't apply to us anymore. Okay, but it might if somebody wanted to make something. And if you're in a chemistry lab, that would be the case. Okay. So then it goes on to discuss other characteristics that have to do with polarity and membrane penetration, et cetera, et cetera. And these get taken into account in specific ways. The one feature that will come up as we look at the structures is that a number of these buffers are what are called zwitterionic, just like glutamic acid. Arginine are zwitterions. A lot of these buffers are zwitterions. They have multiple charges, okay? And so it increases the ways that they can interact with the solution, with the molecules in the solution, the macromolecules and the other components in the solution. And this, in principle, is thought of as a detriment, but sometimes it's actually helpful. I'll give you examples of that later in the course when we cover some papers on tubulin and microtubule assembly. Okay, so now the method section of this paper discusses a lot of other issues, but then it gets into, get out of the way here, then it gets into starting materials that they buy, measurement of acid dissociation constant. So they actually, once they purified this stuff, they actually had to measure the pKs. They get into metal binding, okay? In other words, there can be reactions between the buffer and some kind of a metal. And so part of the issue is how many reactions are there, okay? And so this is showing you that there are basic forms, protonated forms, and metal complexes, and that the metal complexes can interact with the whatever the buffer is, but there can also be free metal. And so you could write down equilibrium constants for the protonation, you could write down equilibrium constants for the metal binding. And then what's shown down here are kind of overall reactions between the metals in the various forms, okay? And then it's reduced if you assume something about being at the midpoint of the titration. And so the metal binding becomes an issue in certain kinds of biological systems. And so what we're looking for is simply knowing if there's a problem. Okay, so let me go back over here. Okay, so I put in this PowerPoint all of those different points. Okay, and then I put in the PowerPoint a summary of that chapter, that paragraph right afterwards that has to do with other kinds of properties. So this is basically a summary of that list. Okay, and then it discusses what these things are that we're going to be looking at. Okay, and so now I copy pasted that table so that we can talk about the table. So this is a list of the buffers that they studied, the buffers that they synthesized, the buffers that they did for comparison. Okay, and so buffers called MES and pipes, BIS and Bicene, a number of these are basically new. There's some newer ones that aren't on this. The primary ones that I'm familiar with are MES and pipes because they're used in a lot of the cytoskeleton research field, in the tubulin field. And you can see what the structures of these things look like. You can see that they're zwitterions, so that they have multiple charges. You can see that there are multiple protonatable groups on some of them. Okay, so Pipes can actually have a double negative charge. Mess tends to only have a single negative charge. And you can see what the groups are, and then you can see what all of the pKs are. Remember the prescription here. You want pKs that are around 6 to 8. Okay, and so here are the pKs. And so if I want to work around pH 6.9, this is a good buffer to be working around pH 6.9 because its pK is at 6.8. Okay. Mess is not as good at 6.9. It can work because it's within 1 pH unit. Okay. But if you wanted to do a range of buffers between 6.5 and 7.5, you could use this at the lower pHs and you could use the pipes at the higher pHs, etc. Okay. Here's another feature that we talked about. This has to do with solubility. Okay. And it's talking about a saturated solution at 0 degrees. And so it says that at 0 degrees, mess has a solubility of 0.65 molar. Okay, so it's relatively soluble, and that's something that's great for a stock. I usually make up 400 millimolar stocks of mess. Notice it doesn't give a solubility here for pipes because at 0 degrees, pipes is generally not very soluble. It's interesting that it would list this. This is very pH dependent for pipes. It's not as pH dependent for mess at all. Okay, and so they sort of leave that out. And then you can see a whole series of the others. You know, so some things you can go up to 4 molar, 3 molar. Okay, so this is considered sort of reasonable, although it's not necessarily ideal. I think you know that you can make phosphate up to be really highly concentrated. As a mixture, you can certainly get up to things in excess of 1 molar. Generally speaking, you work in the 100, 150 millimolar phosphate range, okay, or less, okay, generally as low as 10 millimolar. Okay, so solubility becomes part of the limiting factor. They make it out to be just stocks. I'm going to tell you that in biophysics, you often vary concentrations over a fairly wide range, so you study concentration effects. We will talk about this later on. We will talk about the phenomena, let me try to write up here, of something called osmolites. Okay, so things that tend to be at really high concentrations and have impact on macromolecular systems that are sort of interesting. We tend to study osmolites because they are stabilizers. And so they might be useful as stabilizers. I will mention this when we do this lecture, because we're going to do a lecture on osmolites and something called macromolecular crowding. But biology often uses high concentrations of stuff as preservatives. So, for example, one of the stories that I can tell you is that if you were to go to a desert, okay, Death Valley, it's 120 degrees, and it hasn't rained in 5 years, okay? And it's just basically dirt and sand, okay? And mostly dead stuff. Maybe there's some cactus. And then it surprisingly rains one day, okay? It rains a couple inches. Within a few days, there are flowers everywhere. Plants sticking up, flowers everywhere. And the question is, where the hell did they come from? And the answer is, well, there are seeds there. Seeds that sat in 120 degree weather for 5 years and didn't degrade, didn't go bad. How and why? Well, because their DNA was encased in some high concentration of an osmolite, high concentrations of reagents that preserved the DNA. It didn't just dry out and die. It held on to some water, but it had a high concentration of something in the seed that preserved it. And then as soon as you put a little drop of water on it, boom, it rehydrated, reactivated all of the DNA, reactivated enough enzymes to resynthesize, and suddenly flowers grow, okay? The other place you hear about this is in sharks. Sharks swim around in the ocean. There's predators, obviously. But they actually have really high concentrations of things like urea and things like betaine, betaine is what it's called, another way of saying the word, in their bloodstream. They're sort of protective. It sort of stabilizes a lot of the structures in their cell structure. Okay, but a lot of organisms have high concentrations of stuff that are protective. Okay, and so this solubility issue is not just a buffer issue. It has to do with other sort of biological features that we'll talk about through this course. We use things like urea and guanidine for denaturation, but we use things at higher concentrations for other kinds of phenomena that have to do with protective effects. So let me give you one other example. Lots of antibodies. So you're familiar with this because we're all familiar with vaccines. The vaccines that were taken for COVID are messenger RNA that then produces a coat protein that then stimulates antibody production. But the treatment that people have been getting, apparently it's not working for Omicron, but there's a treatment that people are getting where they get an antibody infusion. Okay, they get something like 10 mils of a mixture of 2 antibodies. So it's generally, let me write that down up here, 10 mils. It's usually 2 antibodies, each at 60 mg per mil. Okay, so do the math here. That's 600 mgs of one antibody and 600 mgs of the other antibody. These are antibodies to COVID, and they help treat the COVID infection. Okay. These antibodies are usually preserved in some kind of a buffer that maintains their activity. And many of these antibodies will have in them some high concentration, 1 to 200 millimolar arginine. Okay, because arginine has these unusual properties that it helps preserve protein structure. Okay, and so this would be an example of an osmolite that is helping to preserve an antibody. The point here is that these antibodies that I'm describing to you, or the antibodies that all kinds of people take for other kinds of illnesses, all kinds of inflammatory disease, Crohn's disease, etc., usually are not frozen. They usually sit on a bench for something like a year, usually in a cold box, but they could in fact be at room temperature. And they are preserved simply because the structure of these antibodies was chosen to be very stable, and the buffer that they're in is also preservative. So this is another example of an osmolite. Okay, so that's an aside about this one column. Here, this column has to do with temperature. Okay, so this pK right here, 6.8, is usually reported as the pK at 20 degrees. But as you change the temperature, the pK actually changes by a little bit. And you can see here it's changing by 0.0085. Okay, it's getting smaller as you raise the pH. Okay, so if you make up a solution at 20 degrees and then you cool it down, the pH will no longer be what it was on the bench at 20 degrees. If you heat it up to 37 degrees or higher, the pH won't be the same. It'll change, and it'll change by this amount. Okay. All right. And so this delta pK is a feature of each buffer. You can see the numbers vary by quite a bit. Okay. In tests, it's 0.02. In glycol amide, it's 0.029, etc. Okay. And so you have to be aware of the fact that when you make up a buffer at 20 or 25 degrees, generally our labs are 25 degrees, it's actually not the pH that you're going to get at your 37-degree culture or at 4 degrees in your cold box. Okay. And so you sort of have to be aware of that fact. If the stability of your system depends on the pH, then you have to adjust the pH at room temperature to be the right pH under the conditions at which you're going to work. All right. It's going to change, and it's going to potentially change a lot. There's an assignment already out there assignment 2, it's on the website in Canvas. The problem that I'm giving you is PCR. When you do a PCR experiment, when you do an experiment and you're trying to monitor how much of a message is in your sample, you raise the temperature to 90 degrees or something. And then you drop the temperature to 55 for reannealing, and then you do something back to room temperature, and then you cycle again. Every temperature is at a different pH. Does that matter for the system? Presumably that's been worked out by somebody so that the enzymes that are being used will work in those pHs. But that's one of the things you sort of need to know, that the pH can vary widely when you go up to 55 degrees or you go up to 90 degrees. Okay. Each one of those is a different pH. You may not know about it, you may not care about it, but when your system doesn't work while you're developing a method, this may be one of the reasons that it doesn't work. In choosing a buffer, you have to start with an initial condition that gives you a pH that works at the other conditions where you're doing the chemistry that you're doing, okay? And then the final thing that's in this table has to do with metal binding, okay? So notice here that the buffers might bind magnesium. A lot of them, it says negligible. Some of them gives a number, and these are log Ks. So these are generally very small numbers. Even 2.5 or 4 is 10 to the 4th, 10 to the 5th. But it says that these buffers can bind magnesium and calcium. They can bind manganese, something that most of you never use, but it can be used as a magnesium substitute and for some systems, and it can bind copper. Okay, and again, something that most people will never use, but some systems require copper. Okay, and so you'd like to know if the copper that you're adding is actually being sequestered by the buffer, and there's actually no free copper, or there's very little free manganese. Okay, you would sort of like to know that, because if your system required magnesium ATP to function and there was no free magnesium, then your system would fail. Okay, and so you have to be aware of how much free magnesium there is or how much free calcium there is in the system that you're working on. And so they report all the metal binding properties. Most of them are negligible, not all of them. Okay, and so it's something that you sort of have to be aware of. Okay, so the paper then discusses this, and again, I think this is a copy paste. It discusses that knowing what the buffering capacity is in the pH region that you're working is a critical aspect. It basically has to do with what fraction is protonated, deprotonated, and how effective the buffer is. And then for certain kinds of systems, there may be side effects. The side effects might be metal binding. Okay. The side effects might be inhibition. It might inhibit. Okay. Inhibition, it might inhibit the reaction or the chemistry. So for example, I worked on tubulin for a very long time, and tubulin uses magnesium GTP. It's inhibited by calcium. Calcium will make a GTP complex, and in principle, calcium GTP will substitute for magnesium GTP in a lot of systems. For actin, for example, actin filament assembly, either one of these works with actin, okay? But calcium will inhibit microtubule assembly because there's a calcium binding site on tubulin that's inhibitory. So only magnesium GTP will work with tubulin, okay? The calcium binding site is not at the ATP site or the GTP site, it's somewhere else in the protein. And so you sort of have to know what these side effects might be, what these inhibitory effects might be. The paper focuses initially on phosphate, okay? Lots of ATP dependent systems are phosphate inhibited. That's why often phosphate is a bad buffer for a lot of those systems, okay? Notice the acid or the base may have different side effects. So different protonated forms may be activating and maybe inhibiting, and you sort of have to know that. Okay. Comparing different buffers doesn't work unless they actually buffer in the pH region that you're working with. Okay, I think the example I mentioned in the book is they say comparing tris and mess or tris and phosphate doesn't work because these have completely different pKs. And so they don't have similar buffering capacity. And so it's not really a fair comparison. You really should only compare buffers that have similar buffering capacity. Okay. And then there's this business about when you have Zwitter ions and there are large ionic strength effects, they may be big side effects. So for example, pipes and mess are very stimulatory for microtubule assembly, and it has something to do with the high concentrations we use, but it also has to do with the charge effects, and it helps drive microtubule assembly. So early on in the tubulin field, using pipes was a big factor in the discovery of conditions that led to microtubule assembly. Okay, so let me get out of this for a moment. I'm not going to save any of those things. You'll have a copy of them here. Okay, so let me go down in this paper and see if there's something else I want to talk about. So the rest of the paper, and I'm not going to go through all of these examples, these guys work on a particular enzyme system. And the reason they did this, okay, the reason they did this, Cindy never showed up, okay. The reason they did this, because their particular system having to do with a particular enzyme system that they worked on, or enzyme systems, they wanted to know something about the buffer effects. They wanted to know something about how the high concentration impacted the chemistry that they were interested in. And so they were comparing different buffers in different plots here to show you, okay, what the chemistry was and under various conditions, okay? And that was sort of the point of their work. It became useful in other papers that they published, and then this described the various conditions that they worked on. Incidentally, this paper comes from a group at Michigan State, and John Hostler actually knew all of these people when they were doing this work, actually. Around that time, he knew all of those people, who those people were. Okay, and so they're showing different kinds of reactions and what the various buffers do to the different kinds of reactions. Notice this unusual activity where it peaks at low concentrations and becomes inhibitory at high concentrations. If that were happening, this would be something that you sort of need to know. You're going to get a big effect at 100 millimolar, and you're going to be inhibiting at 300 millimolar, and it would be kind of useful to you to know that the problem wasn't with your protein or your enzyme, the problem was with your buffer. Okay, and so this is why you tend to work with different kinds of buffers. And then there was this summary of different kind of mitochondrial proteins. This is why John knew these people, because he was already working on mitochondria in those days. Okay, and so different kind of reactions and what their relative effect was on different kinds of reactions under different conditions. Okay, this example, for example, you can see that the rate of a reaction, oxygen uptake, was dramatically enhanced in the presence of HEPs and inhibited in the presence of tris and phosphate. Okay, and so these are the kinds of things that you are sort of need to be aware of in terms of why you're choosing different kinds of buffers. And this is really just, without going into too many details, that's sort of the gist of what this paper is about. Okay, so let me bury that. So the other paper that we're going to talk about has to do with this magnesium ATP condition. So let me pull that paper up here. Okay, so this is a paper by Storer, Cornish, Bowden. These are some Brits, University of Birmingham. Cornish, Bowden was very active in this kind of work in the 60s and 70s. And the question is, if your system uses magnesium ATP, what is it that you have to be aware of in the ionic state of the buffer you're working on? For exactly the same sorts of reasons. What buffer are you going to use? What are the ionic species in the solution? How does it impact the reactions? And what I'm interested in is magnesium ATP. Under what conditions do I have magnesium ATP? Now, I'd like to point something out to you, okay? And it'll become clear down below. What they've written down here is magnesium ATP -2, okay? And so they're writing this down as though this is a fully deprotonated ATP, and then there's a magnesium complex to it. So it's a +2 and a -4. And that's this species, but that's only one of multiple species. ATP has multiple protonations. And so you could have 1 proton, 2 protons, 3 protons. You can have multiple protons in ATP. Okay, which one binds to your protein? Do they all bind? Do only some of them bind? That's part of the point here, that often the binding site for ATP is a magnesium ATP binding site with a certain protonation state. And that's really what this is getting at. Okay, and so the abstract here is a summary of their conclusions. And the summary of their conclusions basically has to do with the full analysis of what we're about to go into. Okay, and so if you make up a solution, all right, if you make up a solution with an equimolar mixture of ATP and magnesium chloride at pH 7, you will have a mixture of magnesium ATP -2. You'll have some ATP -4 with no magnesium. You'll have some singly protonated. You'll have some free magnesium and free chloride. And you'll have mixtures of singly protonated with magnesium. Okay, you'll have something that has 2 magnesiums bound to it. That's actually a possible reaction. And then you'll have some in solution. There's a solubility of magnesium chloride with a plus. All of these species potentially can be in solution. Some of them are minor, some of them are not. All of this is pH dependent. That's sort of the point here, okay? So let's go ahead and then come back. What they write down here is the entire chemistry. Okay, so what they write down here is all of the different reactions that in principle can go on if you make up this mixture for an enzyme assay involving glucokinase. So you know from your biochemistry that glucokinase will use ATP to phosphorylate stuff. The buffer that they're using here is glycoglycine. It will dissociate a proton. Okay, and so there's a binding constant. This is an association constant for the proton to glycoglycine. So there's your buffer. The glycoglycine can actually interact with magnesium, weak affinity, 21 is its binding constant, but nonetheless, it can exist. The magnesium chloride has a really weak binding constant, but it can exist. And then all of these other species involving ATP and magnesium and protons, all of these species in principle can exist. This is the full gamut of the potential chemistry. When we talk about this in biochemistry, we simply say ATP. It's not the truth. ATP exists in protonated forms. ATP exists in magnesium complex forms. Okay, you can see this reaction is 10 to the 4th. That seems weak. This one is 10 to 8. So this is also about 10 to the 4th. It's about 8.5 times 10 to the 3rd. But in millimolar concentrations, that's appreciable amounts of stuff that's potentially being formed. Okay. And so what they're doing in this paper is solving the reactions, all of these equilibria, as a function of pH. Okay, and then they're plotting the concentrations of all of these things for different kinds of mixtures. They're showing you 5 millimolar ATP, 6 millimolar magnesium, 100 millimolar potassium. And then they're going to plot what all of the different species are at different pHs between 3 and 10. And then the question will be, what is it that you're interested in? They're primarily interested in this species 4, and under what conditions are they going to get the best results? Okay. Now, the paper is actually a methods paper having to do with solving multiple equilibria. And the example that they give you is that imagine you have a mixture of R, S, and T, okay? 3 components that can associate to form an RR, an RS, an ST, and an RST complex. So there are 4 equilibria from 3 components, all right? And then they say, well, one of the ways of solving this problem is to write down the total concentration of R. And there would be free R, and there would be an RR species, and there would be 2 Rs because it's a dimer. And then there would be an RS species. There's 1 of those. There would be no R in ST. There would be 1 R in RST. And likewise for S, and likewise for T. And then you can write this down as an equilibrium between the different species. And then that can be plugged into a series of equations where you say, what's the total amount of AI? And there'll be the free, plus there'll be this chemistry. And the chemistry will depend upon the equilibrium constant. Okay, and so species J will be the concentration constant times the product of whatever the reactants are. So for example, there'll be 2 Rs if you're making R2, and so it will be R squared with the chemistry. And then you can plug this into this equation up above here. And this will be the family of curves that you have to solve. And then they do a substitution down here to equate this to individual concentrations of species. So notice this A here. Let me write this down. This A here is being plugged in. A sub I times A is equal to A sub I, but then it's times AI, the big total concentration, divided by AI. Okay, so all they're really doing here is they're substituting 1 AI over AI, and they're using this as sort of this repetitive sort of way of calculating new estimates of concentrations. And so this iterative technique is you plug in a guess, and then you use that to calculate the next concentration. And they show an example of that here, where if you had R, S, and T, you plug in a number, and then you use that to solve the next equation. And then you use that to solve the next equation for AS, and you use that to solve the next equation for AT. And you do this again and again and again. And then eventually this converges to so many decimal points. And then you end up with the final concentrations, depending on the equilibrium constants, et cetera. So they're constructing a mathematical way of solving multiple equilibria at the same time. And here we're showing 3 components and 4 species. 4 complexes. It's actually 5 species. It's actually 8 species. I'm sorry. It's 1, 2, 3, 4, 5, 6, 7 species, because it's the 3 free and the 4 complexes. And so they're going to use the same thing here, where we have 10 reactions. And we have all of these species that are involved in the reaction. So they're going to use this same method. And what they're going to do is they're going to fix the pH. So any reaction here that uses proton will have a fixed pH, and then they'll solve that equilibrium with that fixed pH for that particular example. And then they'll solve this whole series of reactions and then plug a number in for each of them at a pH, and then they'll plot the whole thing out. So that's what's going on here. Okay, now let's see if I can, is it better in my PowerPoint? Let's see if it's better in my PowerPoint. It's not better. What I'm looking for is the table at the bottom. So notice species 1 is ATP deprotonated. And so that's species 4, excuse me, that's species 1, which is right down here. You can see that it's got a low concentration. This is all millimolar on this scale. So 1 is a low concentration. 2 is singly protonated, that's 2. And so you can see 2 peaks, and then it comes down. So it peaks at around pH 4, and then it comes down. Species 3 is doubly protonated. So species 3, where the hell is species 3? Oh, here it is. It comes down, and as species 3 comes down, species 2 comes up. Okay, so as you raise the pH, this is deprotonated, and you make that species 2. Species 4 is the species that these guys are interested in. So it's basically deprotonated ATP complexed with magnesium. All right. You probably don't think of this as existing in solution, but that's the species that is often in solution. And you can see here it comes up and peaks above pH 4, excuse me, pH 7. So that's the predominant species that they're interested in, because that's the species that binds to the enzyme that they're interested in. Okay. And then species 5 is binding to the protonated form. And so that's species 5, which also goes through a peak. So you can see that it comes up with species 2. The magnesium binds to it, but then it goes away as the species 2 goes away. So as this comes up, the magnesium complex form comes up. And as this goes down, this form goes down. Okay. And then there's 2 protons at species 6, which is way down here. It hardly ever exists. And then species 7, which hardly ever exists. Species 8 has to do with a potassium form, which hardly ever exists. Free magnesium is species 9. You can see there's a lot of free magnesium until it's mostly complexed in this species that we're interested in, the magnesium ATP form. Okay. And then magnesium chloride is species 10. You can see there's actually a lot of it, millimolar amounts of it, that kind of flatten out at around, I don't know if that's about 0.3 millimolar, something like that. Okay. And so the question is if you were aware of the fact that magnesium ATP bound to your enzyme, you would have to actually be aware of the fact that it binds in a certain protonated state complex to the magnesium. Okay. And so the question is how do you make up your buffer? Notice that this is done as 5 millimolar ATP and 6 millimolar magnesium. So there's always an excess of magnesium in their buffer. And the paper describes why they do that, because they basically want to push the reaction. You would normally say, well, if I just add 1 millimolar and 1 millimolar, I'll have magnesium ATP. And what this figure is showing you is that's not true. You will not have what you think you have. So here's 1 millimolar magnesium in this table. And then here's increasing amounts of ATP. And you can see at that's millimolar. So that's 1 micromole. No, that's 10. Is that 10 micromole? That's 100 micromolar, 10 micromolar. Yeah, that's 1 micromolar ATP. And this is plotting, this is listing the percentage of ATP present as magnesium ATP. Okay, so this is just percentage. You can see here, it's under these conditions, it's in the range of 80 to 90%, but you can see how it changes across here with increasing concentrations of ATP. Okay, this is excess of magnesium. So if you have micromolar, you're adding 1 millimolar. If you have millimolar, you're adding 2 millimolar. If you're adding 10 millimolar, you're adding 11 millimolar magnesium. If you're adding 100, you're adding 101. Okay, so it's in the excess that you'll get these numbers. But if you do something that has no excess, so here's no excess, okay, you just put in micromolar 10, 100, 1, 10, 100, you can see at these lower numbers, 1 millimolar with 1 millimolar magnesium, you actually only have 65% of the ATP in a magnesium complex. When you have an excess, you get 85%. And if you have a higher excess, you get more. Okay, now notice how this falls off. This falls off probably because other species involving magnesium probably become predominant, and it steals the magnesium from the ATP. Okay, and so you want to be in some reasonable region in the middle here where you have slightly excess of magnesium to be able to get into a region where 80 to 90% of your solution is magnesium ATP, the thing that you think actually binds to your enzyme, the thing that actually gives you activity. So appreciate that. 85% bound, that means by definition, only 85% of your enzyme is active. Okay? You will never get 100% activity at any time. Okay. And that's only if you put in an excess of magnesium over the ATP. If you don't put in an excess, you'll only have 65% activity. So again, what is it you're trying to study? Well, I'm trying to study the activity of this enzyme, glycokinase. Oh, gee, it doesn't seem to be very active. There must be something wrong with my enzyme. Or you just don't know how to make up a buffer. Okay, it's that simple, all right? That you want to make sure that the buffer and the reagents that you're using are not the limiting reason that your enzyme is not completely active. Notice it's never 100%, right? So you're never going to have 100% activity. We do calculations in biochemistry where we plot enzyme activity. And notice we often do something that looks like this. And we look at something having to do with activity, percent activity. And we often have peaked activity curves that look like this, right? And we say that this enzyme is active in this region, often because there might be some functional group, one that gets deprotonated and one that gets protonated, okay? And so we get a peaked activity because we need both a deprotonated and a protonated form of the functional group in the active site, okay? But this percent activity here is what we measure. It's never 100% because you never have 100% of deprotonated and protonated. And then there's this additional factor that I don't ever mention in the class when I'm talking about this. The additional factor is that the active site, the cofactor, is never more than 80, 85, 90% bound. So the actual top, the top activity is way up here someplace. How much protein you actually have, how much enzyme you have, what fraction you're observing, but this is all some smaller number than the total because of cofactor binding, because of protonated and deprotonated groups in the active site. Okay, and so this is what the paper is partially about. Let's maximize our conditions. Now, the way this often works in the actual literature is that people report something and other people mimic what they do, okay? And they say, under so-and-so's conditions, this is what we get, okay? And so let me find a place to write this. Okay, so my conditions for microtubules were often 2 millimolar magnesium. It was actually magnesium sulfate, okay? And usually 100 millimolar pipes, usually 1 millimolar GTP, okay? And then we always had something like 2 millimolar EGTA. And the reason we had GTTA in there was because it chelated calcium. And if there was any calcium in the buffer, that would be inhibitory. And so I would drive the calcium concentration down to 0 because the EGTA would sequester the calcium. In the early 70s, when people were working out the purification of tubulin and conditions for polymerizing microtubules, nothing would work until somebody realized that contaminated calcium was being inhibited, was an inhibitor. And they added EGTA and suddenly the polymerizations worked, okay? We'll talk about this later in the course because I'm going to cover some tubulin papers. A number of years ago, this is way back in the, the late 80s, early 90s, somebody repeated this measurement and they used manganese sulfate. And what they found out was that manganese sulfate didn't work as well as magnesium sulfate. And they reported that, okay? Long paper, varying the manganese concentration. They might have actually used manganese chloride instead of manganese sulfate. And so the paper was published in Biochemistry, okay? The problem is they used EGTA because it's a component of a microtubule buffer. And manganese is sequestered by EGTA, okay? So it makes a complex. Their buffer had no manganese in it. They were getting a lower effect because they had no manganese and didn't know that EGTA bound manganese. I repeated their experiments without EGTA or in the presence of EDTA because this reaction doesn't go very well. And manganese is actually better than magnesium. It makes a tighter GTP complex. It binds to tubulin with higher affinity. It was a great cofactor for microtubule assembly. They just got it wrong because they didn't know that one of their reagents was inhibitory. It was inhibitory because it was sequestering the manganese. All right. That's the kind of thing that you just don't want to publish because it's sort of trivial. We won't call it stupid, but it's certainly trivial, and you shouldn't have done it. You should have sort of done your due diligence and just looked it up. Okay. Now, they plot here the effectiveness of maintaining a linear concentration of magnesium in constant excess over ATP. And they're simply showing that as they increase the ATP concentrations under pH 8 conditions, the amount of magnesium ATP grows in a linear fashion. So this is the various concentrations of all those species. And that's the thing they want to study. All right. Later in the course, we will make plots. We will make plots that are essentially log-log plots. And we will vary the amount of binding of something. So, for example, magnesium binding, okay, to some kind of reaction, and this might be an equilibrium constant, for example, microtubule assembly, and we will be asking what the dependence is on the magnesium concentration, and often dependence will be linear in a log-log scale, and the slope of this line will end up being a factor that we call delta N, and it has to do with how much magnesium binds to tubulin during microtubule assembly. And the answer is 1 approximately. The number usually comes out to be around 0.9, okay? But the answer is usually around 1, okay? Because magnesium binds to tubulin as a complex, and it ends up being sort of a 1-to-1 factor. There may be other magnesiums that bind, but this tends to be what it is that you're measuring with this. Okay. But if your concentration of the ATP or the concentration of the magnesium doesn't actually vary linearly with the magnesium concentration that you add to the buffer, then this curve will generally not give you a true report because the solubility is different. If this curve sort of fell off like this, then you couldn't expect the response to be linear if the concentration wasn't linear. And so you would have to sort of know stuff about the buffer to be able to do this, okay? Now, this is pH 8 down here. And the reason they chose pH 8 has to do with the fact that at pH 8 right here, this is the region in the curve where it flattens out. So they knew if they wanted to do their experiments under certain conditions, they wanted to tend to work around pH 8 if they wanted a linear response from the ATP, okay? If they worked at pH 6, then they knew they were not going to get a linear response, and they were going to get different answers depending on how their buffer conditions changed. pH 7, it would also still not be linear. It would begin to flatten out a little bit. It would be better, but it certainly wouldn't be as good as 8. Now, a lot of systems, a lot of biological systems, a lot of macromolecules don't like pH 8. Tubulin hates pH 8. Above 7.5, tubulin starts precipitating. It does not like things below 6. It starts precipitating. And so your system will often dictate the pH over which you can work. All right. But here's another criteria. Here's a cofactor that you need in your system to study the enzymology. And so you need to be working in this region where things are linear in magnesium ATP concentration. All right. So that's the point of this. Okay, let me see what else I have down here that I want to talk about. And the answer is nothing. And let me come here. And that's what I had. Those are the things that I had in the, that's the stuff that I was supposed to put in here, and I didn't put it in there so I could refer to it. Okay, so that's what we were referring to there. Okay, so what we've covered we reiterated what a buffering region is. And then I introduced you to a paper in 1966 that introduced a whole family of new buffers. They are generally referred to now as Good's buffers because the first author of the paper, his name was Good. Okay. There's one of the terms in there is expense, relatively inexpensive buffer. I tend to buy mess and pipes by the kilo. So you buy 1,000 grams. I think today a kilo of pipes costs $500. It used to be in the 200s, then it went to the 300s. So it's no longer inexpensive. And the same for mess, okay? Pipes is still the cheap way to go, all right, if you can. But this is just sort of the economics of how things have changed over the years. So it mentioned a whole number of features having to do with temperature dependence, having to do with solubility, having to do with metal binding, okay? And so these are things that you sort of have to be aware of, whatever your system might be. And there are sources for this kind of information. If you want to know about metal binding, you go back and you look at the Goods paper. This stuff is usually accessible on websites where you buy the buffers. It'll usually give you information like this. Again, most of you aren't going to work with copper. It will only be people like John, because copper is in his system, that he might be working with copper. Most of you will never work with manganese, okay? I work with it. A lot of people work with it because it's a substitute for ATP. There are very few enzymes that use manganese. There are some, of course. Calcium, magnesium are much more common. And there are concerns that you have to have about them. There's a lot of disagreement about using magnesium chloride and magnesium sulfate. The sulfate adds something to the ionic strength that helps with the reactivity that I've already mentioned, having to do with the buffer as well. Okay. And then we went through the magnesium ATP paper, and part of that is a math problem showing you how you would solve the problem. Okay. Again, appreciate this is a 1966 phenomena. There were no iPhones in 1966. There were no PCs in 1966. There were barely computers in 66. If you went to the computer center in 1966, you were working on what was called a mainframe, usually an IBM 360. You literally carried a stack of cards. It was a box about that big, and it had these what looked like, and what's an analogy? They're kind of long, thin cards that you would punch instructions on, and you would define equations. So for solving problems, and that formula that's in the beginning of that magnesium ATP paper, the solution to that was coded in some way on some kind of a mainframe or some kind of a computer, which would generally be one on somebody's campus. Obviously, we've gotten far away from that kind of stuff, but that's why that kind of stuff was useful at the time. It was an iterative method. You could have solved it in other different kinds of methods that have to do with matrices. We'll talk about this a little bit next week when we do statistics and linear least squares. One of the things that I've never done in this class, I've thought about it for years, but I've never done it, was to introduce computer programming and kind of introduce that as part of the course. I've never done it for various reasons. Most of you don't have a need for computer programming and probably never will have a need for computer programming. In my world, we code, we develop software. I'm going to show you some examples of that in sort of the middle of the course. I'm going to have you download some software and do some calculations with them. We're going to talk about it a little bit when we do the hydropathy topic. I'm going to have you go to certain websites and use some software that's on various websites. That's generally your exposure to this kind of stuff. It would be a useful skill, but it takes a lot of time and effort. And the people I know who teach a similar course, they end up spending the first month of the course teaching computer programming. This is usually done in the presence of Mathematica. A lot of people are using Python as the vehicle. Again, for various reasons, I've made a decision many years ago to not do this with you. Early on, all of my coding was done in Fortran. We went through a phase of using Python. We went through a phase of using something called Turbo. My collaborators today all still use Fortran, although I have my collaborators who also code with Python. So it would be a useful skill, but for whatever reason, I have decided that it's something that I don't want to teach you guys. If you have an interest in it, you should just go and take a self-taught course, probably in Python, because that seems to be the most versatile one today. I was watching a video yesterday from a conference called PyCom, Python conference, where they were discussing next week's topic, which has to do with statistics. It's something that I do in lab all the time, and I'll show you this when we get to the software I'm going to tell you about in another month or so. Okay, so that's it. So I'll post that PowerPoint out there, but it really contains nothing that isn't already in the paper. And then there's an assignment. It's assignment 2. It's in Canvas. You should be able to see it. It's just simple applications. It's the PCR experiment. And what else is it? I forget what it is. Here it is, assignment number 2. Oh, I asked you to do math. Okay, so I asked you in the goods paper. Sorry, somebody's making noise in the back. Oh, you can't see me because I'm not sharing the screen. Sorry. Multitasking. So here's the assignment. You should be able to see it in Canvas. It mentions the equations 5 and 6 in the goods paper. So it's basically converting these 4 introductory equations. Goodness, come on, get up there. To the summary equation, the overall equilibria. Okay, so it's combining these 4 equations to derive this more complicated equation, what the overall metal binding constant is, in addition to all of the protonated states, and then an assumption that reduces the whole thing to 6, okay? I'm doing that with you because a lot of this course involves deriving formulas. So we're going to get into some of that next week. We're going to get into a lot of algebra when we do thermodynamics. We're going to get into a lot of algebra when we get into binding. And so I want you to practice your algebra, okay? The biggest example of this will be when we derive the equations for Monod, Wyman, and Changeau. Remember, I discussed this in biochemistry. Ahmed, I can show that to you later if you want to see those lectures, but you'll see it when we do this. Oxygen affinity for hemoglobin, T in our state, and you get these two competing equilibria, one low affinity, one high affinity. Well, that was actually derived by somebody, Monod, Wyman, and Changeau in the early 60s. And there's a lot of algebra that goes into it. Okay, and so I'm going to go through all of that algebra then, but I will also then ask you to kind of take something and explain it in a more complicated way, derive the next step of something. Or I think what I actually usually ask you to do is I skip a step. That step is usually about 10 steps, and I ask you to fill in the blank. Okay, and I'm just basically trying to hone your algebra mathematics skills. A lot of this course, a lot of biophysics is mathematics. A lot of it is physics, a lot of it is chemistry, some of it is biology, but basically it would be helpful for you to just practice. And so that question that I'm asking you is practice. Okay. All right, so I'm done for today. All right, so you'll see ahead that there's already another lecture out there for next week. It involves, I should have that posted. I'm going to just double check to make sure I have what I want out there. Yeah, so those are, there's chapter 3 and chapter 6 from that book. It's called Error Analysis PDF and Least Squares PDF. Those are scanned copies from a book. The book is some version of this book, which is on statistics. It's written by this guy, Bevington. There's always a second author because Bevington died many years ago, and they keep republishing his book with his name on it. I guess he's not getting royalties anymore. So there are various versions of it in different sizes and different colors, but these chapters are things that I want you to, things that you will need to know. And I'll make it clear next week what it is that you clearly need to know. And then there'll be another problem set. And I think the problem set may already be posted, or I will post it soon. Okay. So you're all being very quiet as I talk. Maybe this material today is very straightforward. The papers were in front of you, and all I was really doing was presenting the papers to you. I was trying to drive home some practical features about buffers, buffering effects, things you have to worry about with various buffers. As I've already said, you're working in a lab that tells you what to do. Okay. My lab very often plays with conditions. We play with salt concentrations, we play with pH, we play with temperature, we play with the kind of buffer that we're using. And we publish papers based upon that kind of stuff, right? We're looking at the pH dependence, the temperature dependence, the salt dependence, etc. But I think it's helpful for you to know this. We're not getting real practical because, again, I think most of you don't even make up your own buffers. You buy them. Or if you buy them, you know how to use a pH meter. But that's another issue and another skill that we could talk about if need be. All right. So before I sign off and stop the recording, and I'm presuming that it really is recording still. Yes, it's still recording. Do you have any questions?
Dr. Correa, I think I need one more day to submit
That's
assignment.
fine. Some other people have asked for more time, so that's okay.
Yeah, yeah. So
Okay.
I'm still working on the last question, so
Okay,
I'll wrap it up.
no
tomorrow.
problem.
Thank you.
Any other questions? Any other comments?
Mm.
Anybody? Okay. So I'll stop the recording. It'll speak to you when I do that. Probably it'll say